Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Listeria Monocytogenes, or simply L. monocytogenes, is a gram-positive bacterium that causes listeriosis. Listeria was identified by a Scottish doctor, Dr. James Hunter Harvey-Pierre, and named after British surgeon Joseph Lister. It is also known as monocytogenes because when it was injected in rabbits, their monocyte levels rose. L. monocytogenes is a rod-shaped bacterium with a thick peptidoglycan cell wall. When gram-stained, it absorbs purple dye and becomes a gram-positive bacterium. It is catalase-positive and oxidase-negative, which implies it generates catalase, but not oxidase. L. monocytogenes is a facultative intracellular pathogen, which means it may dwell either outside or within its host cells. It does not produce spores and is facultatively anaerobic, which means it can live in both aerobic and anaerobic settings. When L. monocytogenes is cultured on blood agar media, the colonies produce beta hemolysis, commonly known as full hemolysis. This is because it creates toxins known as beta hemolysins, which hydrolyze hemoglobin within red blood cells, resulting in translucent yellow byproducts. L. monocytogenes is a motile bacterium with a unique movement pattern that is affected by both location and temperature. In an external environment, this bacteria travels by beating its flagella, resulting in a distinctive tumbling motion. However, this is only achievable at temperatures of 37 degrees Celsius or lower. That's because Fla-A, the gene that codes for flagellin, the structural protein that makes up the flagella, is downregulated when the temperature increases over 37 degrees Celsius. So above 37 degrees, there's reduced production of flagellin proteins, meaning no flagella being made, rendering L. monocytogenes non-motile. L. monocytogenes travels in an intracellular environment via actin-based motility. It begins with the bacteria creating a protein known as actin assembly-inducing protein, or simply ACTA, which attracts tiny actin filaments at one end. As additional actin filaments are recruited and polymerized behind the bacterium's end pole, they propel the bacteria forward like a rocket. L. monocytogenes is a foodborne bacteria, which means that humans become sick when they consume contaminated food, particularly unpasteurized dairy products and cold deli meats, and it is known to persist at extremely low temperatures in the refrigerator. When this bacteria enters the gut, it employs attachment proteins called internalins to bind to the host's receptor proteins, such as cadherin, which is found on goblet cells of the intestinal mucosa. After connecting to the host's cells, this bacterium is gradually absorbed by the cell membrane, which invaginates to create a sac on the inside. The sac then splits from the cell membrane, generating what is known as an internalization vacuole. Inside the vacuole, Listeria produces listeriolysin O and bacterial phospholipases, which are enzymes that break down the membrane. This releases the bacterium into the host's cell cytoplasm, where it begins to consume the host's cell resources and nourishment before multiplying into many identical bacterial cells by binary fission, which means that each bacterial cell divides into two identical copies. Interestingly, this is comparable to mitosis. However, the phrase binary fission refers to the division of prokaryotic cells that lack a nucleus. Therefore, certain replication processes differ from mitosis. L. monocytogenes travels around inside the cell utilizing actin-propelled motility, stretching the cell membrane towards a neighboring cell. When Listeria reaches the neighboring cell membrane, it also invaginates, resulting in a double-layered sac. The sac then pinches off within the next cell, forming a double membrane vacuole. Inside this vacuole, Listeria secretes listeriolysin O and phospholipases, allowing them to break free into the cytoplasm and repeat the invasion process in order to infiltrate additional neighboring cells and finally enter the circulation. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. The pathogen travels through the circulation to other organs, causing disseminated listeriosis. This is especially frequent in those with weakened immune systems, such as newborns, the elderly, pregnant women, and adults with immunocompromising illnesses, including diabetes, cancer, or HIV infection. Under these conditions, Listeria can develop a liver abscess or spread to the brain via the blood-brain barrier, causing meningitis, which is prevalent in newborns, the elderly, and diabetic people. The worst-case scenario in pregnant women is when germs cross the placenta and reach the fetus. This may lead to a spontaneous abortion or stillbirth. If the fetus lives to term, it may be born with newborn listeriosis, which can result in sepsis and meningitis. When listeriosis affects only the intestinal mucosa, it causes symptoms similar to gastroenteritis, such as low-grade fever, diarrhea, and vomiting. Disseminated listeriosis causes a high temperature, muscular soreness, and other symptoms that vary depending on the organs involved. Abdominal discomfort and jaundice are common signs of liver abscesses, while meningitis causes headaches, stiff neck, and altered mental state. Neonatal listeriosis is often characterized by fever, respiratory distress, and a distinctive skin rash that is widely dispersed. Granulomatosis infantiseptica is a condition marked by tiny, pale nodules. Cultures from a biological sample are used to diagnose a variety of conditions, including gastroenteritis, bacteremia, meningitis, and transplacental infections. Imaging investigations, such as an ultrasound or a CT scan, can help to detect liver abscesses. When listeriosis is restricted to the gastrointestinal system, therapy often consists of supportive measures such as fluids and relaxation. However, disseminated listeriosis is treated with intravenous ampicillin and gentamicin. Meropenem is an effective alternative to these antibiotics for those who are allergic to ampicillin or who are pregnant. To summarize, Listeria Monocytogenes is a gram-positive, rod-shaped bacterium with facultative anaerobic and beta-hemolytic properties. It is likewise a catalase-positive, oxidase-negative, facultative intracellular pathogen. Listeria possesses tumbling motility in the extracellular environment and actin-based motility within the cells. It most usually causes listeriosis in newborns, immunocompromised people, and the elderly. Listeriosis can cause low-grade fever, diarrhea, and vomiting, which can progress to high-grade fever, jaundice, and altered mental state in cases of disseminated listeriosis. Listeriosis is diagnosed by cultures, and imaging tests such as a CT scan can assist detect liver abscesses. In pregnant women or those who are allergic to ampicillin, treatment is with a combination of ampicillin and gentamicin, or with meropenem alone. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!